about all sorts of sequencing of biogas producing microbial communities. Thanks very much.
Right, thank you. Yeah, I'm going to present this, this project where we look at biogas producing communities basically. So we do metagenome, metatranscriptome, single cell sequencing. We look at the metaproteome. We are at a very early stage of the project, but I think I can give at least some ideas how we are going to analyze the data we get. So why are we interested in these biogas producing communities? There is a transformation of the energy supply system in Germany. Basically after the meltdown, the nuclear meltdown in Fukushima, Germany decided to shut down all the nuclear power plants in Germany. And they want to do this by 2022, and they want to replace the nuclear power by renewable energies, basically. That's a goal. And as of last year, or actually it's 2 years ago already, already about 12% of the power in Germany is actually from renewable energies, which is actually more than the nuclear power already. When you look at the renewable energies, then like 64% of energy is from biomass actually, and then a certain percentage is from the biogas, which is basically methane gas. So how do these plants look like? This is an overview of such a plant. So you take the biomass, you put this in these anaerobic digesters. There are microbes in there, they degrade the biomass, and then at some point the methanogens produce methane. And then you can either use the methane for heating, or you can actually burn it on site and produce electricity. And whatever is left in these digesters, you can actually put back onto your on the land and use it as a fertilizer. And obviously there are some advantages of using this because it's a clean and renewable energy source. It has a favorable CO2 balance, and you can actually store and transport it. And the storage and transport infrastructure for methane gas does exist already. The limitations are in Germany are that the cultivable land in Germany is limited. So you're competing with like other, well, food, for instance. And that's why we are looking at this process, because we want to optimize this process, or at least first understand it, and then the ultimate goal is to actually optimize the process. So that's a study design. We are looking at, actually there is a larger project looking at, I think, 30 different biogas plants. They are monitoring this over 2 years, taking metadata like how much methane is produced, looking at pH and whatever you can measure in these fermenters. And we picked 4 of them, and we sequenced the 16S just to see what's in there. We sequenced metagenomics, DNA basically, to see to look at the functional potential of these communities. We look at the transcriptome to see who is actually active. And then because this is an anaerobic process, we want to look at single cell genomes, because most of these microbes in these fermenters they are actually very hard to culture, especially the archaea who produce the methane gas, right? Okay, so the whole sequencing is actually done by JGI. This is one project of the user program Eddie mentioned earlier. So if we look at the 16S data first, this is how it looks like for 4 of these plants. We, as I mentioned, we have more data, but I'm only showing the metagenome data for 4 plants here. We do this in replicates. So these are, as you can see, well, you can see that these are replicates, technical replicates actually. And what you do see is that there is some difference in the different fermenters. And well, you have to note that the y-axis actually starts at 50%. So most of the community are actually, or the color doesn't really show up here, but are firmicutes. Like over 80% in this community are firmicutes, and only a very small proportion are actually the archaea, which produce the methane gas at the end. So it's only, let's say, 3 to 6% maybe of the whole community. Okay, but we are not only interested in the taxonomic profile, but also in the genes and the function of the genes. So this is the raw data we got back from JGI a couple of months ago. Again, these are replicates, so you see two different sequencing data sets basically for all of these plants. So altogether we have 400 gigabases of metagenome of DNA, and we have almost 300 gigabases of RNA. And the first thing we do is actually we assemble it, because we want to look at the genes and we want to map the RNA reads back to the genes to get a measure of which genes are highly transcribed. And when you assemble it, you have to come up with some parameters for the assembly. So we try different assemblies, but we cannot really run many of these assemblies because the data sets are just too large. And this is just an example of running the Ray assembler. The nice thing about Ray is that it's actually distributed. You can run it on a whole cluster. And these are two different assemblies with two different k-mer lengths. So the blue bars are for k-mer of 21, the red ones for k-mer of 41. And as you can see, the total assembly size we get is around like 1 gigabase, 1.2 gigabases. That doesn't really change much between the different assemblies, but what's changing is the N50, for instance. So you get longer contigs, basically. And that's nice because we are looking for genes, so we want to get long contigs and predict genes on this. You get, or you see the largest contig is growing. Of course, the number of contigs is going down. But when you look at the total number of genes we predict on these, then you see that the assembly is still pretty fragmented, because here, for this one, for instance, we predict 5 million genes for only 1.4 gigabases, which is way too much because you would expect to see like 1 gene per kilobase, right? So that seems that the second assembly is actually a little bit better. 3 million genes for 1.4 gigabases. It's still not, well, perfect, but we decided to go on with this assembly basically. And then we also looked at the complete genes, like how many of the genes are predicted as complete genes, and you can see that for the longer k-mers, more of the genes are actually predicted as full-length genes, but still it's only like 20% of the genes which are predicted full-length. Okay, and then you can start looking at differences between the different communities. And what we did is we not only assembled these samples separately, but we did a combined assembly of all the data sets. And then we can map back the genes, map back the reads of the different samples. So here we're looking at contigs basically. And what you see is the coverage for each of the contigs. So this is the coverage of the replicate for the first sample. This is the first replicate, this is the second replicate. And what you can see is that the replicates actually produce very similar results, right? Each of these dots in this plot is a contig. The contigs are colored by the GC content, right? The idea is, well, what we actually want to do is to color this by taxonomy, for instance, but we haven't done this yet. But you already see some clusters showing up where like contigs which have similar coverage have the same GC, for instance. So now what happens if you compare different samples? This is sample 1 compared to sample 2, and what you see is that some organisms show up in both of these samples, some only occur in one of these, or they occur with very high coverage in one of these and pretty low coverage in the other one. Like this one here has only 100x coverage in one sample, but 1000x coverage in the other, right? But again, you see these clusters of contigs with similar GC content. And then you can look at different combinations. This is plant 1 compared to plant 4, and when you look at this, you see that there's actually something going on. What you don't really know is if this cluster here is this one or this one or this one. This is a little bit difficult to look at. So just to get an idea what's going on. This is an animation. This is like morphing one coverage to the other. So down here is it's basically morphing the coverage from one sample to the coverage of the other sample. And what you see is that these clusters actually stay together, right? They don't get separated, which means this is probably the same genome, but it's just changing its coverage in one of the one of these samples. So that's very nice, and you can actually use this kind of information for binning, for instance. And I think Chris will talk about these kind of approaches tomorrow. Okay, and then again, I said we're interested in function, and these are the main processes that are going on in these plants. So it's a hydrolysis of the biomass. You have to break down the biomass basically. Then there's acetogenesis going on, acetogenesis, and at the end there's methane produced. And of course, we are mostly interested in the methane at the end. So let's look at the transcriptome. Again, for the transcriptome, we're interested in highly expressed genes. But what you cannot really do is use RPKM values to compare the different things because there are different abundances in different organisms, of course. So what we plot here is the DNA coverage again for each of these genes now. So each data point in here is a gene, a predicted gene. And we plot the DNA coverage of each of these genes versus the RNA coverage. And what you want to see is something that's up here, highly transcribed and maybe, well, high coverage. Not necessarily. You can look at things that have low coverage but high transcription, but you can see there's some sort of correlation again.
Those predicted genes are based on the context. You said this each dot is one predicted.
Well, yeah, on this plot, each dot is a gene. On the previous plot, each dot was a was a context, right? But here you're looking at genes now.
Based on the context.
Based on the context, yes. So we assemble it, we have context, we run a gene prediction, and then we get these genes, right? And then we map the DNA reads back to the genes and the RNA genes back to the genes. And again, you can start looking at taxonomy, for instance. These are methanomicrobia, and I don't know. There might be two different clusters here in this group of organisms. So maybe there's one with higher coverage than this one. But what we are really interested in are the methane-producing genes. And it turns out that the highly transcribed genes are actually genes which catalyze the last step in the methanogenesis. So these MCR genes are these red dots now. And this is actually interesting because if you remember the 16S plots, the 16S profile, the methanogens make only up like 3% of the whole community. But it seems that this 3% is basically, well, I don't know, just a couple of genomes because we get very high coverage for these genomes. So you don't have a whole community of methanogens, but it seems that it's very limited number of archaea. Okay, we also do metaproteome analysis. That's another approach just to see what kind of expression you have. And we were actually able to identify, like on this one here, well, we identified 120 of these proteins on the gel. And what's interesting is if you cut out this spot and then you run your mass spec and try to identify it using the SwissProt database, we could only identify like 26% of the genes. But if we use our assembly, we go up to like 67%. That's another reason why we actually do the assembly, because now we have a powerful, like a database where we have proteins and can compare to. And of course, the proteins we find again are involved in the methanogenesis again. Okay, and then we also want to do single-cell genomics. I don't really have data for this, but I wanted to show you the pipeline we are using. So again, this approach is very similar to what Eddie described for the microbial dark matter project. What we do is we get basically two plates of cells. You amplify the genome using this MDA, multiple displacement amplification method. You identify the cells by 16S sequencing, and then you pick the ones you are interested in. Unfortunately, none of these single cells is a methanogen because they are they don't like oxygen, right? So we are not really sure at what point they already die. It might be early on in the preparation of the sample. It might be later on when we actually, or the JGI is actually sorting the cells. We don't really know. But the most important ones, or the ones we are most interested in, are unfortunately not part of this 700 cells. So how does the assembly for single cells look like? This is the assembly pipeline we developed at the JGI. The problem with the assembly is basically this step here, because this step introduces a tremendous bias in the coverage of the genome. And when you look at this is a genome here, and this is the coverage. The coverage, you have all these peaks of very high coverage. So the coverage goes up to, in this case, like I don't know, 1600x coverage, right? And all these peaks make the assembly really difficult. So I will show you an approach how we actually deal with it. But another very important thing is the quality control, because you want to make sure that you don't pick up like free floating DNA, or you don't have two cells in the same well after sorting and so on. So what you do is a quality control. You can just look at the GC content of the reads, for instance, or the contigs after assembly. And as you can see in this case, it seems that there are actually two different cells in here with different GC content. You can color this by taxonomy, or you can use other approaches. This is a tetranucleotide-based identification of contaminants. So this actually are two cells which look pretty clean. If you look at contaminated samples, and it looks like this, it turns out that there are actually two different clusters. So that's a way to detect the contamination. Okay, and then you start assembling. And as I said, we developed this pipeline. For the pipeline, Tanja Wojcik at the JGI sequenced 8 single-cell genomes from these genomes where we had a reference for, so we can compare to the reference. As you can see, they have different GC content and so on. And one problem is, as I said, the bias and the coverage. The other problem is that you don't pick up the whole genome. You just have holes in the genome, right? And you will never be able to assemble it just by looking at one single cell. Okay, so what do we do for the assembly? It's a very easy approach, but it seems to work. So when you look at the coverage again, so in this case you get up to 15,000x coverage actually for some of these regions. We just use a K-mer approach to cut the high coverage. So we just throw away all the K-mers that have very high coverage, and after that, the coverage basically looks like this, right? And then you can use just a standard assembler for assembling it. And how does it look for the benchmark? So that's what we call the gold standard. The gold standard would be 5x coverage, let's say. These are the 3 different genomes again, colored in the different colors, 8 copies, 8 single cells for each of them. And this is the best you can get, right? By just mapping it to the reference. And you can see for the different single cells, it's already very different, right? The total assembly size for these differ a lot. The number of contigs, this is the number of contigs. You don't expect to get one contig, NG50, largest contig, and so on. And if you just run Velvet, for instance, it looks pretty bad. The total assembly size is, sort of okay, but the number of contigs is just, it's way too high, right? So you get pieces of the genomes, basically. And 50 is very low, largest contig, very short, and so on. It makes a lot of mistakes, like underlined, this is a number of underlined bases, indels, mismatches, and so on. You can do this with other assemblers. There's a single cell version of Velvet that actually doesn't do much better, not at all. I mean, look at the total assembly size. It's nothing gets assembled. IDBAUD looks pretty good already. like it generates large, well, the number of contigs is, well, it still generates lots of contigs, total assembly size, but the NG50 is still pretty low. And then there's a special single cell assembler, Spades, which does a pretty good job too. And down here is our pipeline, which is actually comparable to Spades, except that we don't make that many mistakes. But that's actually an earlier version of Spades, so the Spades team made improvements to that, and I think Spades is pretty good now. It's basically the standard for single cell assembly. The other thing you can do is, if you want to capture the whole genome, you can start pooling single cells, right? So whenever you see a 16S and you think it's the same genome, you can pool. You just pool the data sets, right? And if you do this for these genomes, you can see that for E. coli, for instance, if you look at the single cell, this is just one cell here. What you get back from this one cell is between like 65% and 90% of the genome. For other genomes, it's worse. When you pool 2 of these data sets, the coverage gets much better. If you pool 3 of them, it even gets better, and so on. So if you happen to have 6, 7, 8 single cells from the same genome, you can just pool the data sets, and at the end you hopefully get a complete genome back. So, but what if you don't have multiple copies of this genome? That's another approach a PhD student in my group is working on. If you have a corresponding metagenome, then the genome of this single cell is present in the metagenome, right? So what you can do is you have some, well, what you have is some overlap of the reads of a single cell, and then something that's not in the metagenome maybe, but most of the genome should be present in the metagenome. So what you can do is you just go into the metagenome, you recruit the reads, and then do another assembly. And the nice thing about it is you're also normalizing the coverage, basically. So up here you see the assembly for one single cell. These are the contigs, basically. This is up here as a reference. This is the coverage in the single cell reads. If we use the single cell reads and go into the genome, the metagenome, and just recruit all the reads from the metagenome, you get, well, an even coverage. And if you assemble these reads now, you get a single contig instead of like 4 of them. But what you also get is you can reconstruct the low covered regions, right? Basically, you only need one single read from the single cell to recruit like hundreds of reads of the metagenome, and now you are able to assemble this part of the genome which you couldn't assemble just in the single cell assembly. So we actually reconstruct more of these genomes using this approach of co-assembling the single cell and the metagenome. Yeah, that's basically it. So as a conclusion, I would say, okay, you do want to assemble your metagenomes, but you have to be careful, right? You make sure you don't misassembly, you don't do misassemblies, look at the contigs and so on. But I think it's worth assembling it and not only looking at the single reads. And also, if you can do single cell genomes, then it's a very nice approach of like coming from both sides, basically, and then because these methods really nicely complement each other, and you are able to construct like whole genomes out of your metagenome. Okay, and with that, I would like to end. This is like most of the work is actually done by Andreas Bremges, PhD student, and these are the biologists working with the samples and actually extracting DNA and RNA and so on. Okay, thanks.
In this recruitment of extra reads. So if that's easy, then it would suggest that you don't have too much concern about picking up stuff that's closely related but actually doesn't belong to the same organism. So, I mean, how easy is that? Or is it only easy under some circumstances?
Well, if you have many closely related strains, then it's probably difficult. It really depends on the community in the metagenome. So we have seen examples where we can actually where it's really easy, right? Because this particular single cell, this genome, has only one, representative basically, right? So basically, the recruitment we do is based on a k-mer approach. That means we are really fast in doing it. So we look at almost perfect matches, right? And we tried this for complete genomes. So Andreas, he downloaded all completely sequenced genomes and then just did a comparison of how many false positives you actually pick up. And the false positives are basically only very, very related genomes, right? So it seems to work.
I was just wondering, has anybody succeeded in getting a single cell genome from a methanogen?
No, I don't know. Probably not. I don't know of any.
It seems like a fundamentally challenging thing to do, right?
It is. So, people are actually trying to culture it, and it seems that this is enormously difficult because everything has to be anaerobic, basically.
Yeah, no, we're
So the guess is that, like, the whole preparation of your sample has to be done in anaerobic conditions, and then maybe even the sorting, right? Because maybe the cells lyse whenever they are exposed to oxygen already. Yeah.
For the k-mers, are you taking gaps or mismatches into account, or is just like one-to-one match?
For the recruitment?
Yeah.
We look at all the k-mers in the reads, and then we basically require many k-mers, let's say. So it's like a Cougar kind of distance you're using. So the k-mers are perfect, right? But you don't want to find like all of the k-mers, but like almost all of them, let's say, for a read.
What are K? What is size of K?
Here the k is, I think, 21 maybe. Yeah.
So we want to test Malbec for single cell sequencing because it's apparently giving a more even coverage over the genome. Have you thought about that or any experiences?
Well, I only saw the paper, and probably it does, but I think you cannot use it for high throughput, right?
No, I think we have done controls with Malbec versus, I guess, with
the MDA.
MDA.
Yep.
And Steve Quake has also done it, and it hasn't looked as good.
Okay, in terms of
In terms of coverage, in terms of being able to assemble it, and so I think there are some controlled studies which have suggested that.
Okay.
I have a quick question. So if you look at the single cell sequences and then you kind of look at the metagenome, you can start to maybe get an idea of how much of that makes up for the metagenome, yeah? So, and as you start to collect more of those single cells, you'll get an idea of proportions of the amount of the metagenome coming from each of the cells. Have you thought much about, how much does it, because you're using the metagenome to fill in the single cell. But
Yes.
the other way around, if you're looking at how much is being made up by pieces, do you have any ideas or estimates based on that?
I'm not really sure if I understand the question.
Okay. Well, maybe I'll ask another time. But,
Okay. Okay.
I think I understand what Vince is asking.
Okay.
You know the size of a footprint of a single cell that you've assembled inside the metagenome. Can you estimate, how many other single cells would it take to cover the whole metagenome?
Oh, that's what you mean. Well, how do you know? That's the question is how many different species are there,
Yeah.
right? And what we do, what we get from the 16S analysis are only like, maybe you can down to the genus level or something like this. And then even then you have, like if you look at the OTUs, we probably have like 500 OTUs in there, which doesn't mean we have 500 different genomes, but many more, right? So that's a difficult question. And you, when you do the sorting, excuse me, when you do the sorting, you probably pick up the most abundant ones with higher chance, right? So I don't think you get the full picture.
okay, well, thank you very much.